Next topic, genetic transmission in viruses. In living organisms, the genetic material is well packaged and is transmitted from parent to progeny in a highly ordered and precisely programmed manner. In multicellular organisms, genetic transmission is of two kinds, lateral and vertical. Lateral transmission always occurs from parent cells to daughter cells, whereas vertical transmission occurs from parent organisms to daughter organisms. In unicellular organisms, there is no such distinction because full-fledged organisms are only glorified cells. The genetic information inherited by an organism is phenotypically expressed through the synthesis of specific proteins. In sexual and parasexual mechanisms, genes are transferred from parent to progeny as reshuffled packets. This is mainly due to an orderly programmed process of gene shuffling known as genetic recombination. Recombination ensures that each new progeny will have a particular gene combination a little different from the parent combination. Yet it remains or retains the species-specific traits since it conserves the minimum quota of the basic gene combinations and the basic traits of the species. The mechanisms of genetic transmission are different in viruses, bacteria, and eukaryotes. So, viral genome and genetic transmission in viruses. Viruses are infectious intracellular replicate parasites with subcellular level of organization. They are devoid of protoplasm, cells, cell organelles, enzymes, and the molecular machineries for energy metabolism and protein synthesis. They are extremely simple, ultra-microscopic organisms formed of only viral proteins and DNA or RNA. They lack the powers for metabolism, so they depend on the host cell for the raw materials and enzymes for essential functions. Thus, viruses can multiply and undergo mutations only in living host cells. Outside the host cell, they are simply inert particles. First, we are going to learn about the genetic transmission in bacteriophages. Bacteriophages or bacterial viruses are the viruses which parasitize bacteria. They were found by British scientist Stuart in 1915 and later described in detail by the French scientist de Herelle in 1970. They are widely distributed in nature, which are especially found in large abundance in entire bacteria which inhabit the intestine of man and other animals. They are especially adapted for adsorption and penetration through the rigid cell wall of bacteria. There are several variants of phages. Usually each variety is specific to a particular species or strain of bacteria. Bacteriophages fall under four major groups, namely single-stranded DNA or ssDNA phages, double-stranded DNA dsDNA phages, single-stranded RNA ssRNA phages, and double-stranded RNA like dsRNA phages. Now let's learn about typical structure of DNA phage. Google image for that. T4 bacteriophage, commonly called the T4 coli phage, can be taken as a typical bacteriophage. It is a double-stranded DNA phage with binal, cubical, and helical symmetry that parasitizes the intestinal bacterium Escherichia coli, hence the name T4 coli phage. It has five structural parts, namely head, neck, like head to tail connector, that is neck, tail region, base plate, and tail fibers. Head is shaped like an icosahedron. It is an elongated bipyramidal and hexagonal prism which is about 95 nm long and 65 nm in diameter. The central core of the head is formed of a linear and double-stranded DNA molecule associated with some cations, several peptides, and 3 internal proteins. The DNA is characteristic of having a hydroxymethyl cytosine instead of cytosine. The core DNA is encased by a capsid formed of about 2000 capsomeres or protein subunits. Head tail connector or neck is a very short region in between the head and neck. It consists of a collar or a disc and several fiber-like whiskers. Tail is a narrow cylindrical part about 80 nm long and 18 nm wide. It has a central helical and hollow core or tube through which the viral DNA passes to the host cell during infection. The core is surrounded by a helical and contractile sheath of proteins. It is formed of 144 protein subunits arranged in 24 rings of 6 subunits each. The sheath is connected to the collar basically and to the base plate terminally. Base plate or end plate is a hexagonal plate with a pin or spike at each corner. It is terminally connected to the tail fiber. Tail fibers are 6 in number, arising from 6 corners of base plate. They are used for attachment to the base host cell wall during infection. Each of them is nearly 130 nm long and 2 nm wide. Each fiber has two portions or half fibers, proximal and terminal. Proximal half fiber is attached to the base plate, and the terminal one is used for attachment to a receptor site in the cell wall of the host. So tail fibers have the proper power to recognize specific receptor sites in the cell wall of the host. Then viral genome. Viral genome is extremely diverse. It consists of single-stranded or double-stranded RNA or DNA. Viral genome may be composed of one of the following, like single-stranded RNA chain, double-stranded RNA helix, single-stranded and linear DNA, single-stranded and circular DNA, double-stranded and linear DNA, double-stranded and circular DNA, and double-stranded DNA with covalent bonds. Covalently bound terminal proteins. Since viruses are able to replicate only inside their host cells, viral genome must be organized in such a way that it might be easily recognized by the host cell. So also the viral genome code must be matched so that the rest of the host of organism and the control signals of gene expression must be somewhat similar in both. Viral genome is very small with only much limited store of genetic information, so it can code only for a few proteins. Consequently, it cannot replicate and phenotypically express all by itself a lot of the necessary enzymes and proteins factors. Therefore, a virus depends upon the molecular mechanisms and the cellular machinery of its host for genetic transmission and expression. To accomplish this, vaccines take control of the molecular machinery of the host cell to synthesize their nucleic acid and coat proteins, and also for the formation of their capsid. DNA viruses and RNA viruses employ different mechanisms for gene transfer and gene expression. Then, genetic transmission in DNA viruses. The mechanism of gene transfer is different in different kinds of DNA viruses. Based on the difference, DNA viruses are classified into lytic viruses, lysogenic viruses, and tumorogenic viruses. Lytic viruses produce many progeny viruses in the host cell and ultimately cause the lysis or disintegration of the host cell. So they are also called virulent viruses. Lysogenic viruses live inside the host cell without producing viral particles and without causing the lysis of the host cell. So they are also called non-virulent or temperate viruses. Most lysogenic viruses undergo lytic cycle also under certain circumstances. So they have a dual method of genetic transmission. Through an open strategy, that is lytic cycle, they produce the maximum possible copies of viral genome as rapidly as possible. Through the alternative closed strategy like lysogenic cycle, they integrate the viral genome with the host cell genome and inherit it as a part of the host genome. Tumor-causing viruses abnormally alter the physiology and morphology of the host cell, make the cell cancerous, and thereby induce tumor formation in multicellular organisms. T4 and T7 bacteriophages, which infect the bacterium Escherichia coli, are lytic viruses. Lambda phages, which also infect E. coli, is a lysogenic virus. Rous sarcoma virus or RSV, found in chicken, is a tumor-causing virus. The method of gene transfer in phages is determined by the nature of the transcriptional regulation of the phage genes. In lysogenic transmission, phage genes are expressed by the interaction between a phage repressor and a phage operator. In the lytic method, there is a cascade of transcriptional controls in which successive sets of genes are successively turned on at specific times. Each set contains a regulator gene that brings about the transcription of the next set. First, we are going to learn gene transfer in lytic or virulent viruses. Gene transfer in the T4 lytic phage is an extensively studied phenomenon. In this case, the virus first adsorbs to specific surface receptors on the bacterial cell wall with the help of its tail fiber. At the point of this attachment, the base bacterial cell wall dissolves and an opening is formed by the action of the viral enzyme lysozyme. Through this opening, the viral DNA is injected into the bacterial cytoplasm by the contraction of the tail region. This may be called viral infection of the brain's bacterium. Little is known about the transfer of DNA into the host cell in the case of tailless virus. Still, it is presumed that the phage capsid breaks open, releasing the phage DNA onto the bacterial cell wall. From there, it gets into the cytoplasm by lysozyme activity. Following infection, the phage DNA takes command of the genetic mechanism of the host cell. During this, it acts as a master, and bacterial DNA serves as a slave. Master and slave hypothesis. Soon the bacterial DNA loses its ability to replicate and transcribe. Eventually, it disintegrates into nucleotide units, which may be used for the replication and transcription of the phage DNA. Now, the phage DNA undergoes replication, transcription, and translation, making use of the molecular machinery of the host cell. During its replication, the phage DNA directs the synthesis of replication system to make identical copies of it. It is accomplished either by the synthesis of phage-specific polymerases or by the phage-specific modification of bacterial polymerases. In both cases, several bacterial replication proteins may be employed. T4 lytic phage, host bacterium, etc. are involved. The transcription of phage DNA is almost always initiated by bacterial RNA polymerase. But after the first transcription event, either the bacterial polymerase is modified to recognize phage promoters or a phage-specific RNA polymerase is synthesized. Transcription is regulated in such a way that phage proteins are synthesized as and when required. Usually, there is clear difference in the timing of the synthesis of phage-specific enzymes and structural coat proteins. This temporal difference is established by appropriately regulating the timing of transcription. Once the replication, transcription, and translation of the phage DNA are over, many hundred copies of it would be formed and sufficient amounts of phage proteins would be synthesized. The phage proteins include structural proteins, catalytic proteins, and maturation proteins. Structural proteins are the coat proteins which form the structural units of the capsid. Catalytic proteins are the enzymes which mediate the assembling of viral particles. Maturation proteins are the catalytic proteins which bring about the chemical modification and functional maturation of the phage DNA for packaging it with coat proteins. The coat proteins now assemble around each copy of the phage DNA. This gives rise to new progeny of mature phages. Shortly afterwards, the phages release the enzyme isozyme endolysin. It disrupts the bacterial cell wall and disintegrates or lyses the bacterial cell. This results in the liberation of the progeny phages to the surrounding medium. They may reinfect new bacterial cells to repeat the cycle. Some filamentous phages continuously release their progeny by unfolding the bacterial cell wall without causing serious damage to the bacterium. This process is called budding. In such cases, viral particles may be produced continuously over long periods of time. The successive events in the lytic cycle are regulated by three sets of viral genes, namely early genes, class 1 genes, second genes, middle genes or class 2 genes, and late genes, class 3 genes. Early genes constitute the first 20% of linear viral DNA. In about 4 to 8 minutes after infection, they undergo transcription with the help of the RNA polymerase of the host cell and from viral mRNA. This mRNA soon gets cleaved into segments. Each segment undergoes translation on host ribosomes and produce a viral protein. Second genes constitute the next 20% of viral DNA. They are of two major groups. The first group is concerned with the production of the enzyme which bring about the disintegration of the bacterial DNA to provide free nucleotides for the synthesis of phage DNA. The second group is concerned with the production of enzymes which bring about the actual synthesis of new phage DNA. Second genes undergo transcription and translation in about 6 to 10 minutes after infection. Late genes constitute the remaining 60% of the phage DNA. They are concerned with the synthesis of capsid proteins and also with the packaging of phage DNA within capsid proteins to form new phages. One of the late genes is especially involved in the synthesis of the viral enzyme isozyme, which causes the lysis of the bacterial cell. Late genes become active in about 5 minutes after the activity of the second gene is over. Viral infection of the bacterium is always accompanied by genetic recombination. In most cases, more than one strain of virus enter the bacterium. Since their genetic composition is different, there is every possibility for recombination. This viral recombination is peculiar in that some DNA molecules can take part in many recombinations in the same infection cycle. The details of the molecular events of viral recombination are not quite clear. Still, it is assumed that genetic recombination in viruses involves the formation of joints with gaps and overlaps. The gaps may be filled in by the activity of appropriate polymerases, and the overlaps may be degraded by endonucleases. Sometimes members of the same strain of virus enter the bacterium. In such cases, recombination takes place by breakage and heteroduplex formation. Since the genetic information in all the DNA is almost the same in this case, marked changes in the phenotypic manifestation of the recombinant DNA may not be observed. Next topic, genetic transmission in lysogenic or temperate virus. More than 90% of the phages are lysogenic. Among them, the lambda phage is the best studied one. In lysogenic phages, progeny virus will not be produced and the host bacterium will survive and divide indefinitely for many generations. After that, the lysogenic cycle may come to a halt and lytic cycle may follow. During this, progeny phages are produced and then released by the lysis of the host cell. There are two types of lysogenic cycle. In the first type, the phage DNA gets closely incorporated with the bacterial DNA. This is the component type and is found in the lambda phage. In the second type, the phage DNA behaves like a bacterial plasmid and it replicates independently inside the bacterium without getting incorporated with the bacterial DNA. This is less common or very rare and is found in the P1 phage, which infects E. coli. A bacteria which harbors a complete set of lysogenic and lytic phage genes is called a lysogen. The transformation of a normal bacterium to a lysogen by viral infection is called lysogenization. The process by which phage genome get incorporated with bacterial genome is called insertion or integration. The close association between the viral and bacterial genomes leading to the repeated replication of the viral DNA as a part of the bacterial chromosome is called lysogeny. The phage DNA integrated with the bacterial DNA is called prophage. The lysogenic cycle in which the phage DNA gets integrated with the bacterial DNA is completed in the following major steps. The phage at first adsorbs to the surface of the bacterium, then dissolves the bacterium cell wall and lysogenize action, and finally injects its DNA to the bacterial cytoplasm. Soon after injection, the phage DNA undergoes transcription and translation inside the bacterium and produces a repressor protein and an integration enzyme called integrase. This is accomplished by utilizing the molecular machinery and ribosomes of the bacterial cell. Very shortly afterwards, transcription is turned off by the repressor protein with the result that the phage genes get repressed. So the phage DNA soon gets integrated with a specific site of bacterial DNA within the help of integrase. This forms a DNA heteroduplex. In this integrated state, the phage DNA is called prophage. During integration, the enzyme recognizes the specific attachment site in the bacterial chromosome and binds with it. The host bacterium goes on growing and dividing indefinitely with the result that the prophage gets repeatedly replicated as a part of the bacterial chromosome. Thus, the phage DNA is transmitted to several generations of host cells in a very short period of time. This harmonious association between the viral genome and bacterial genome is called lysogeny. Under certain conditions, the prophage gets restricted from the bacterial chromosome. This released the phage DNA from the integrated state is called induction of the phage. The released phage DNA soon takes control of the genetic machinery of the host cell, follows a lytic cycle and causes the lysis of the host cell. During it undergoes replication, transcription and translation, producing multiple copies of viral DNA and coat proteins which get assembled to form progeny phages. Lysogenic bacteria have two characteristics, namely they can promote the induction of the prophage to initiate lytic cycle. They are susceptible or immune to superinfection. They will not be reinfected by another phage which is similar to or identical to the one which has already lysogenized it. Then genetic transmission in tumor-causing viruses. The infection cycle of a tumor-causing virus such as the S14 virus which infects monkeys has two options, namely productive Infection and abortive infection. Productive infection pathway is similar to the lytic cycle in that it involves the production of viral progeny at the expense of the host cell. In abortive infection, the host cell is not lysed. In this case, the viral genome is believed to get integrated with the host cell genome, as in lysogenic cycle. However, unlike in lysogenic phages, this integrated viral genome alters the genetic makeup and physiological functions of the host cell. As a result, the host cell undergoes a series of changes and becomes neoplastic or cancerous. These changes are collectively called transformation. In productive infection cycle, nearly 20% of the viral genome, which constitutes the early genes, undergo transcription inside the host nucleus and produces viral mRNAs. These mRNAs move to cytoplasm, where they get translated to viral proteins on the host cell ribosome. Of these proteins, the so-called T antigen, transformation antigen or tumor antigen, is very important. It is essential for the replication of the viral DNA, transcription of the late viral genes, and activation of the enzymes of the host cell. Several hours later, replication of the viral DNA, the transcription and translation of the late viral genes occur. As a result, many thousand copies of viral DNA and tens of thousands of viral protein molecules are formed. These get assembled in the nucleus of the host cell, forming progeny virus. Shortly afterwards, the host cell dies. In abortive injection or infection cycle, T antigen is synthesized by the activity of early genes, but in the late genes, which code for capsid proteins and the enzymes of viral DNA synthesis, are not expressed. Sometime later, the host cell undergoes repeated division and the resulting daughter cells show altered morphological and physiological features. This is called transformation. Some of the transformed cells may revert to their normal original form and become functional after a few days. So their transformation is called abortive transformation. The others stay on permanently in the altered state. This is called stable transformation. So that's all about this topic of genetic transmission in viruses. We have dealt with the bacteriophage genetic transmission. Thank you for listening.